So what is dependent assortment of alleles? Now this happens when the two characters are passed from parents to offspring as a package. The two characters are passed from the parents to the offspring as a package together. As a whole, the dominant Y and the capital R alleles relevant for yellow color and round seeds, for example, are passed together. As this. They pass together as this unit. Generation after generation. Now this is called dependent assortment of alleles. Now to really understand this properly, once again I show you an example. Let's take our famous example again. Capital Y, capital Y, capital R, capital R. What's this plant? The plant that contains yellow rounded seeds, and it's pure line, crossed with simple Y, simple Y, simple R, simple R. So what's this plant? The green plant with wrinkled seeds. Now as usual, you can produce these gametes. There's no problem with that. But the problem comes after this. Now this exists as one unit. This also exists as one unit. Right? You can't separate them now. Therefore, here only two varieties of gametes are produced. To understand this better, to support you better, I'll show you when dependent assortment of alleles are not present. That is, when independent assortment of alleles are present, how would you come across like this? Now, as you can see, we have got the first F1 generation. Now up to the F1 generation, in independent assortment of alleles and dependent assortment of alleles, it's similar. But after the F1 generation, now when you are giving rise to the F2 generation, there's a difference. Now in dependent assortment of alleles, only you can produce two types of gametes. But here, see how many gametes you can produce. You see, Y can go, capital Y can go with capital R, capital Y can go with simple R, simple Y can go with capital R, simple Y can go with simple R. Did you see the difference? So you get four types of gametes here, right? Don't you? What do you get? You get capital R, capital R, capital Y, capital R, you get capital R, Y, simple R, you get simple Y, capital R, you get simple Y, simple R. So you get four types of gametes here, don't you? This is independent assortment of alleles, but earlier we saw dependent assortment of alleles. Now with that understanding with us, let's see this definition again. What is dependent assortment of alleles? The two characters could be passed from parents to offspring as a package. The dominant Y and the capital R alleles relevant for yellow color and round seeds, for example, are passed together generation after generation. This is called dependent assortment of alleles. Good, so we'll go on to the next one. We did that independent assortment of alleles in our previous video, a short one. So this next definition is one of the most important definitions in genetics, and is frequently asked in past papers. So what's that? Mendel's second law. I'm sorry, we have missed some definitions because we have to discuss Mendel's first law first. So we'll come to those in a moment. So breeding. So what is a test cross? What do you mean by a test cross? Breeding an organism having unknown genotype for a specific dominant trait. Which kind of organism are you breeding now? An organism having an unknown genotype. You don't know what his genotype is. For a specific dominant trait, it's like this. You have a plant. Let's say you have two plants. One plant, right, is actually, let's say I know about the plant, but you don't know about the plant. I know one plant is homozygous dominant. That is, it's very tall, it's homozygous dominant, it has capital T alleles. The other plant, it's also tall. But I know, because I have done experiments, I know, and I have done crosses, I know it is capital T, simple T. But you know that, but somebody else comes in and you're not there. He wants to discover what kind of plant is this. And you tell him, let's say I tell him, this particular plant is homozygous. But I don't know about the other plant. Let's say you're lying, you don't know about the other plant, right? But you know that it's heterozygous, but you're just telling one of your friends, I don't know what this plant is, but it's also tall. And then your friend is suggesting, shall we do a cross with these plants and see? That's the basis of this definition. Try to remember that example. So a breeding an organism having unknown genotype for a specific dominant trait. It's exhibiting that dominant trait though. Breeding an organism having unknown genotype for a specific dominant trait with an organism having homozygous recessive condition for the same specific trait is called a test cross. Breeding an organism having unknown genotype for a specific dominant trait with whom? With an organism having homozygous recessive condition. With an organism having homozygous recessive condition for the same specific trait is called a test cross. And for the same specific trait, if you are having a recessive condition, you take that organism and cross with the organism who is showing dominant characters, but you are not sure about his genotype. It's like this. You are going to take that plant. Now your friend came close to you and you told, I know the other plant is capital T, which is dominant, purely dominant, homozygous dominant, but I don't know about the other plant, but it's still tall. Now you have to take that plant and you have to cross it with a short plant. Now if short is a recessive character, right? If it's a recessive character, it should contain simple T alleles, right? It should have the recessive condition, however. From this cross, you can find out what the unknown plant is, whether it's homozygous dominant or it's heterozygous dominant. I'll show you a cross, then you can understand this better. Let's say we have a tall tree and you told your friend that this is purely dominant and you have done experiments and you know it's a dominant plant, purely tall, right? So let's take that out of the equation and just suppose now we have to think, now this is our tree. We don't know whether it's heterozygous dominant or whether it's homozygous dominant. How can we test whether it's homozygous dominant or heterozygous dominant? It's like this. Suppose we have to think of one possibility first. Let's think it's tall. Homozygous dominant tall. We have to cross it with our other plant. Other plant, we know if it's having the recessive condition, it should have simple T, right? Short plant is short, and that's our tall plant. All right, so If it's tall, the generation that we get is TT, 100%. 100% tall plants, don't we? We get 100% tall plants. Now we'll see if it's heterozygous dominant, what will happen? With heterozygous dominant, we can you come across this. You have capital T, simple T allele, and this common T, simple T allele here. So we are doing a cross. What happens? What you get here is a 50-50 thing. You get capital T, simple T plants, and you get simple T plants. Now these is 50%. This is also 50%. Now if you come across the observation that all the plants you got from the cross in our experiment were tall, then surely it should contain the homozygous dominant alleles. But suppose you got a mixture of these plants, 50-50, then surely we have had a heterozygous dominant plant. So that's the understanding behind this definition. So once again, let's look at this definition. Because genetics, I'm taking more time because these definitions are harder to remember. Right? There's a lot of understanding in this. You can't memorize definitions. I don't know if you all can, but I can never memorize definitions. Try to get into the definitions, understand them, and then you can read over and over again. Then it retains in your head. Breeding an organism, because understanding definitions not only is useful, it's not only useful for just writing structured essay, but also when answering MCQs, right? To get the clear understanding of the subject and also answer certain questions, mathematical questions in genetics. Breeding an organism having unknown genotype for a specific dominant trait with an organism having homozygous recessive condition for the same specific trait is called a test cross. Now you know what a test cross is because we did one test cross, right? It's very easy now. The next one. Now we are going to talk about Mendel's law of segregation. That's Mendel's first law. Mendel's first law of inheritance. Mendel's law of segregation. So what is it? During the formation of gametes, when gametes are formed, during the formation of gametes, alleles for a heritable character. So alleles responsible for a heritable character, right? When the gametes are formed, follow with me, alleles for a heritable character are separated and get into each of the gametes formed. During the formation of gametes, alleles for a heritable character are separated and get into each of the gametes formed. So to understand, it's very simple. Capital T, simple T, you have, let's say, capital T and simple T, they separate as capital T and simple T. So this is the capital T gamete and this is the simple T gamete. That's it. So during the formation of gametes, alleles for a heritable character. So this character should be inherited, should be a heritable character. During the formation of gametes, alleles for a heritable character are separated and get into each of the gametes formed. We saw that now. The next one. What do you mean by a Punnett square? You have drawn Punnett squares, but do you know what it really means? A Punnett square is a graphical representation is a graphical representation of what? Of the possible genotypes. It's a graphical representation of the possible genotypes of an offspring arising from a particular cross or a breeding event. Now you know what a Punnett square is. The Punnett square is a graphical representation of the possible genotypes of an offspring arising from a particular cross or a breeding event. So I hope you can go through the books and see diagrams of Punnett squares and the respective crosses. My main focus here is to give an understanding about the definitions, because if I am to explain about Punnett squares, I would have to explain a cross and show. So I don't think I have time for that. So as you have learned in classes, you can see that there are so many examples of Punnett squares in your book. The next one, we discussed this, didn't we? This part, yeah. So Mendel's second law of independent assortment, alleles separate and pair up independently during the formation of gametes, as a result of which two or more genes assort independently irrespective of each other. Don't think it's hard, just think very simply. Try to remember the examples we discussed in our class today. Within that framework, you can see this definition is already there in those examples. Focus on the main words here. Mendel's second law of independent assortment, says what? What does it say? Alleles separate and they pair up independently, no disturbance, during the formation of gametes, as a result of which, what happens as a result of which two or more genes assort independently, irrespective of each other. Now you saw the yellow rounded seed plants, right? When you saw that the yellow rounded seed plants were crossed with the green wrinkled plants, you saw how the capital Y allele combined with the simple R allele or the capital R allele. Did you see all those? Remember? So how those combined, right? It's independently they can combine. There's no disturbance. Mendel's second law of independent assortment, says alleles can separate and they can pair up independently during the formation of gametes, and as a result of which two or more genes can assort independently irrespective of each other. So look at the examples and read this definition again. You can understand that very clearly then. Always go with the examples in genetics. Now we are coming to a very interesting definition. The multiplication rule or the product rule in probability. This is very simple. When the occurrence of one event does not affect, so that's the crucial part, does not affect the occurrence of another event, the probability of simultaneous occurrence of both events can be obtained. How? By multiplying the probability of one event by the probability of the other event. This is known as the product rule or the multiplication rule in probability. When the occurrence of one event does not affect the occurrence of another event, the probability of the simultaneous occurrence of both events. That means the probability of occurrence of the two events at the same time, it can be obtained, that probability can be obtained by multiplying the probability of one event, that independent event, from the probability of another independent event. Now this is known as the product rule, or the product is or known as the multiplication rule in genetics. I think I need to draw a Punnett square for this. To understand the definition, you need this. Let's take this. Capital TT, I'm not explaining a lot. I'm doing it quickly. You can understand these basic things now. You know what happens here? You get the cross, you get capital T, simple t. This is the parental generation. So parents, gametes, these are the gametes, and you get the F1 generation genotypes, capital T, simple t. Now if you draw a Punnett square, capital T, simple t, if you are crossing the F1 plants among themselves, capital T, simple t. This is the sperm, this is the egg. Look at the box and draw in a standard way. I'm just doing because we don't have much time. So what do you get? Capital T, capital T, capital T, simple t, capital T, simple t, simple t, simple t. Now what does this probability rule tell you? Multiplication rule. To understand this, observe this carefully. Now you know in this F1 generation, suppose you have parents, two parents, each parent is represented as is having this genetic, this genotype. The other parent is having this genotype, for parent 1, parent 2. I have to erase this part. So suppose the probability that each of these parents, now you know the probability of one parent, let's start like that, the probability of one parent having the simple T allele is half, right? Because capital T can be produced from him, simple t can be produced from him, right? So the probability of producing a simple T allele from parent number 1 is half, because he can only produce two types of alleles, right? Half. The probability of producing simple T allele from parent number 2 is also half. Now we are coming to the important part. What's it? Now your father producing, let's say yes, father producing simple T allele in one of his gametes is half. Mother producing simple T allele in her egg is half. Now these two occur in two different organisms and they are independent of each other. They're separate, right? Those two separate events occurring together. So you say, right, I want your father and your mother to have simple T combination to produce a simple T combination. So for that to happen, for these two things to happen together, that means your father making a simple T gamete and your mother producing a simple T gamete, for them to fuse together, that event happening together is multiplied. I hope you understood that. So this is a multiplication of two independent events. So this one independent event and that's another independent event. What's the next definition? Addition rule of probability. The probability that two or more mutually exclusive events. The probability that two or more mutually exclusive events will occur is calculated by adding their individual probabilities. This is known as the addition rule of probability. Probability that two or more mutually exclusive events will occur is calculated by adding their individual probabilities. This is the addition rule of probability. Actually, there should be some words here missing. A rare mistake here. Came across this just now. The probability that any one of, there should be that word, any one of. One of. Probability that any one of two or more events, more mutually exclusive events, will occur is calculated by adding their individual probabilities. This is the addition rule of probability. Let's see an example. We drew a Punnett square earlier. We need that same Punnett square again to clarify the meaning of this addition rule of probability. Remember that example. From that example, you can understand the definition again. So as you can see, we're taking one monohybrid cross. But wrinkled seeds, you remember capital R is the dominant allele representing round seeds. Simple R contributes to form what? Wrinkled seeds. So simple R, those plants produce wrinkled seeds. Capital R, simple R, plants produce round seeds. Now I'll just give you a shortcut to understand this. Now can you see that there are four possibilities here? Four possibilities. So from those four possibilities, the possibility of happening capital R, simple R, when, suppose these are the sperms represented, sperms, these are the eggs. Suppose the probability of getting capital R, simple R from father, capital R from mother to give capital R, simple R heterozygous stage. So what's the probability of this? Probability of that is taken from the product rule we discussed earlier. So go back to that, try to understand that again if you did not, and come to this. So then the probability of getting a simple R allele from the father into the gametes is half. Probability of getting a capital R from mother is also half, because there are only two possibilities, right? Capital R or simple R from mother. So mother, if she gives capital R, that is half. So you get 1/4 of a probability of getting capital R and simple R here. In the same way, getting capital R and simple R, when you get simple R from your mother and capital R from your father, is also 1/4. Calculate and see. Same thing. Is also 1/4. 1/4. So these are two different events, right? So as a totality, as a whole, the probability of getting a heterozygote is 1/4 plus 1/4. Now this is the addition rule that we were talking about. Plus. This is what the addition rule means. There's another way you can take that. In your calculations you can take, now there are 4 units here, right? This is unit number 1, unit number 2, unit number 3, unit number 4. From this you can see 2 common units, capital R, simple r, capital R, simple r, common units. The probability, let's say 4 individuals are formed. From the 4 individuals, the probability of getting heterozygotes is 2 upon 4, because you have 4 and you have 2 in common, heterozygotes, right? That's the probability, that is half. Simple. I hope you understood that easily. Now let's go back to the definition and read it together. The probability that 2 or more mutually exclusive events will occur is calculated by adding their individual probabilities. This is the addition rule of probability. Now, there's another logical part. Try to understand that. It doesn't matter if you don't really understand that. What do you mean by mutual exclusivity? That is when 2 events occur, when 2 mutually exclusive events occur, they cannot occur at the same time. Now, one example is when you toss a coin, it can result in either heads or tails, but not both, right? It can give you either heads or tails, but not both. That's the logic behind mutual exclusive. Don't think too much about it. It might be confusing for you. So just follow that example and remember this definition. The probability that 2 or more mutually exclusive events will occur is calculated by adding their individual probabilities. This is the addition rule of probability. What is a dihybrid test cross? When an individual with dominant traits for 2 characters is crossed with a pure recessive for both characters, it is known as a dihybrid cross. When an individual with dominant traits for 2 characters, this time with dominant traits for 2 characters, is crossed with a pure recessive for both characters, is known as a dihybrid cross. Like, for example, we can take this simple y, simple y, simple r, simple r. This is what? The recessive pure organism. And the one we want to find out where is purely dominant. Who's that? It can be what? It can be capital Y, capital Y, capital R, or it can be capital Y, simple y, capital R, simple r, right? I'm not going to show this test cross. You can go to your book and see that. It's in page 100 of your resource book because it takes a long time. So our main focus here is to understand the definitions, not teaching the entire lesson. The next definition, what is a pedigree chart? I think this came for the exam also once. What is a pedigree chart? A diagrammatic representation of the inheritance of a particular trait. The diagrammatic representation of the inheritance of a particular trait within a given family tree is called a pedigree chart. So together with me, a diagrammatic representation of the inheritance of a particular trait. You are seeing how the trait of your grandfather or grandmother, how the traits of them, it goes through generations to the family tree. A diagrammatic representation of the inheritance of a particular trait within the given family tree is called a pedigree chart. So it's a diagrammatic representation. And what does it show? It shows the inheritance of a particular trait within a given family tree. You have to look at the examples of pedigree charts, then you can understand this better. Now we are coming to a very easy definition. What do you mean by complete dominance? The phenomenon of dominant allele completely masking, we did this actually, completely masking the recessive phenotype, resulting similar phenotypes for both homozygous dominant zygote as well as heterozygous zygote is called complete dominance. The phenomenon, the incident of dominant allele completely, right, 100% completely masking, right, removing the expression, masking the expression of the recessive phenotype, resulting similar phenotypes for both homozygous dominant zygote as well as heterozygous zygote is called complete dominance. So the next one is incomplete dominance. Now this is new. What is incomplete dominance? At heterozygous state, the phenomenon of expressing blend phenotypes from both alleles is called incomplete dominance. So at heterozygous state, the phenomenon of expressing blend phenotypes, this time it's mixed blend phenotypes. That means at capital R simple R stage, let's say in some plant, okay, at capital R simple R stage, what normally happens is, let's say it's red color of the plant, red color happens. You can see red color. But here what happens is pink color like color comes. Let's say some other color comes. Not white. Let's say R represents white. Capital R, simple R represents white. Capital R represents red. Not any of those. Mediate color, intermediate color comes. So at heterozygous state, the phenomenon of expressing blend phenotypes from both alleles is called incomplete dominance. Now once again, I'd love to explain the how you get these things and the examples, but I don't think that this is the suitable occasion for it. You can see in your resource book in page 105, incomplete dominance in Mirabilis jalapa, flower color, right? You can see that. You saw that the red flower and the white flower, when they're crossed, no? You get, when you cross a pure red flower and a pure white flower, what do you get? You get a pink flower. That pink flower is actually, oh, I just show this cross then. I do it quickly. You can see the pure red flower. You represent it like this. In these incomplete dominant examples, you have to show the genotypes like this. This is the second one I have shown, this one, which I've ticked here. It represents white flowers. This represents what I've ticked next is, actually they are the red flowers. Now when you produce gametes, you get CR, CW. That is okay. Up to that it's okay. But when these two fuse, what happens? In the F1 generation, you get CR, capital C, simple C, capital R, capital C, capital R, capital C, simple, capital C, capital W. It's actually this capital W. Okay. So you get this intermediate thing. What is this? This is actually pink in color. Now, this was red. This was white. Now this is pink in color. Right? This is pink in color. I'll just write it down. It's pink in color. So this is not what you expect, right? This is not what you expect normally. Now in incomplete dominance only you get this problem. Okay? So once again, at heterozygous state, the phenomenon of expressing blend phenotypes from both alleles is called incomplete dominance. You can see there was a mixture, right, of a different color is formed because that's intermediate to the two colors. The next definition. What is codominance? Very important definition, codominance. At heterozygous state, expression of both alleles contribute equally. So both alleles contribute equally here. So at heterozygous state, expression of both alleles contribute equally. To what? To the phenotype. Now such a phenomenon is called codominance. At heterozygous state, expression of both alleles contribute equally to the phenotype. Such a phenomenon is called codominance. You can refer to your resource book on behalf of this. So in deciding our blood groups, this is the case. We have IA alleles, IB alleles, simple I alleles, right? Now you know AB blood group, for example, I just tell that IA, IB, right? That's a definite blood group. Because you know a person with AB blood group has A and B carbohydrates on the red blood cells at the same time, right? The two carbohydrates are added to the surface of red blood cells by enzymes encoded by IA and IB alleles of a single gene. So both genes, right? I mean, in that single gene, or both alleles contribute to the same to different functions, but their contributions are equally important, right? Because one allele has to produce carbohydrate A and the other allele has to produce carbohydrate B. So you can't say one is recessive and the other one is dominant, right? Got it. So that's the case. At heterozygous state, expression of both alleles contribute equally to the phenotype, because to the phenotype is decided by the carbohydrates A and carbohydrates B. To the phenotype, such phenomenon is called codominance. Last definition for today, we'll continue it on the other day, is this. Polyallelism. What do you mean by polyallelism? Polyallelism refers to the presence of multiple alleles for a single genetic locus, a phenomenon where certain traits are determined by the combination of more than two types of alleles. Polyallelism refers to the presence of multiple alleles for a single genetic locus, a phenomenon where certain traits are determined by the combination of more than two types of alleles. But this again links into our blood groups. Now, actually remember this statement I'm making. There are three alleles called IA, IB, simple I for a single genetic locus, which at different combinations determine the ABO blood groups in humans. So there are three alleles participating, right, in the same genetic locus, for the same genetic locus. When IA and simple I are together in the same genetic locus, what do you get? Now they are in the population, right? What do you get? You get A blood group. Capital I, IB and simple I. When you get IB and simple I, what do you get? You get B blood group. Simple I, you get O blood group. So they have equal significances, right? So refer to that again. You can see these things in pages 106 in your resource book.